good morning, good afternoon, or good evening, whatever time it is that you are watching this. This is Lisa Whitehead, and we are studying together Biology 205, Anatomy and Physiology 1, and now we're studying Chapter 3, the cellular level of organization. This is the part where you actually feel like you're studying anatomy and physiology because we get into cells, how they function, and this forms the foundation of everything we're going to talk about moving forwards. So keep in mind everything builds on itself, so let's go. The learning outcomes for today's lecture include the following: to compare and contrast somatic and sex cells, to describe how the cell is organized and the function of each organelle in the cell. So this includes the cell or plasma membrane, the cytoplasm, nucleus, microvilli, cilia, centriole, cytoskeleton, ribosomes, flagella, proteasomes, mitochondria, rough endoplasmic reticulum, and smooth endoplasmic reticulum, the Golgi apparatus, lysosomes, and peroxisomes. You'll be able to describe the cell or plasma membrane structure and function. You'll be able to identify the lipids and the proteins that are found in the cell plasma membrane, including phospholipids, cholesterol, transport proteins, as well as receptor proteins. You'll be able to discuss the concepts of hydrophilic and hydrophobic and explain why these are a benefit to the cell's plasma membrane, and to describe the processes and significance of membrane transport mechanisms, and this includes the use of channels, carriers, pumps, and vesicular transport. Also, you'll be able to explain how organelles function together in processes including secretion, and to recognize how organelle malfunction can often affect cell health, to identify the structure and functions of the nucleus and nucleolus, to describe the basic protein synthesis and trace the formation from transcription in the nucleus to translation in the ribosome, then to the rough endoplasmic reticulum, to the Golgi apparatus, and then to transport via a vesicle. You'll be able to describe exocytosis and endocytosis, including phagocytosis and pinocytosis. You'll be able to list the differences between DNA and genes, explain the role of the 5 nucleotides that code for proteins and how many are required to code for one amino acid, and to interpret a strand of nucleotides and determine either the DNA coding sequence, template strand DNA, mRNA codons, or tRNA anticodons. The last learning outcomes for this chapter include to be able to describe the selectively permeable nature of the plasma membrane and explain how the following affects selective permeability: molecular size or shape, electrical charge, and lipid solubility. You'll be able to describe active and passive cellular transport mechanism and what drives them, and this includes simple diffusion, facilitated diffusion, osmosis, primary active transport, secondary active transport, co-transport/symport, and counter-transport/antiport. Also to explain how the following affect diffusion rates: distance, molecule size, temperature, gradient size, and electrical forces. You'll be able to explain what osmosis is and what occurs, and describe the differences between osmolarity and tonicity, and to identify solutions as isotonic, hypotonic, or hypertonic. You'll also be able to compare and contrast interphase, mitosis, and meiosis in somatic and sex cells, and last, you'll be able to explain what happens in the phases of mitosis, including prophase, metaphase, anaphase, telophase, and then at the end, cytokinesis. Before we dive into the chapter content, we're going to discuss a little bit of an introduction first, and then we'll go through the chapter in order as you see here in our outline. So before we delve into cellular biology and anatomy and physiology, let's talk a little bit about basic cell theory first. So we all understand that cells are the building blocks of all organisms, and that they're the smallest possible unit that perform all of the vital physiological functions, right? So all of the different cell types that we have in our body are all going to derive from the division of pre-existing cells, which is fascinating when you really think about it. We'll talk more about this as we get into the lecture. And each cell has the unique ability to maintain homeostasis at its individual cellular level, so it takes care of its own house, if you will. Together, many cells will form a tissue, but for today, we're just going to be talking about cells as individuals. Cytology is literally the study of cells and cell function. And as always, form is going to follow function, right? So the structure follows function, or the anatomy follows physiology is another way to say that. So the image that you're looking at on the right is a cytology preparation called a pap smear, which you've probably heard of before. And what we're looking at there is you can see that there are all different colors of structures because some cells are going to take up different stains a little bit differently. And you're going to see that they're different sizes, that the cells are different sizes, and then further, the nuclei in the center, those dark areas, are varying sizes as well. And this actually means an awful lot, and this is what cytologists look for when they're scanning pap smears to detect cervical cancer. So we'll discuss this more coming up, but this is the very beginning, basic foundation of cellular biology. So what exactly does a cell do? Well, the three main things that it's going to do are going to be metabolism, communication, and reproduction/inheritance, right? So first off, they have to metabolize things. So what does that mean? It means that ultimately they're synthesizing and decomposing. So synthesizing is they are creating molecules, and decomposing, they are breaking down molecules, right? And they do this in order to give themselves enough energy to survive and also to maintain homeostasis. Next, they also communicate with other cells, and that's really kind of a fascinating process. So they have to do this communication with other cells in order to maintain homeostasis for both their own individual self and also for the organism as a whole. And then lastly, they reproduce and they also inherit things, right? So all of our cells in our body are going to come from just two cells, and that is the ova that we obtain from our mother and the spermatid from our father. And so those two cells specifically will come together and, of course, reproduce to form an embryo, then a fetus, and a baby, right? So those individual cells, the ova and the spermatid, those are called haploid in that they only have 23 chromosomes each. So then when those two come together, all of the somatic cells, and soma meaning body, are all the other cells in our body and in our parents' bodies as well. And those are called diploid because they have 46 chromosomes or 23 pairs. And so those 23 pairs are from our parents, right? So one pair is from our mother, from the ova, and one pair is from our father, from the spermatid. This is figure 3-1 from your textbook. It's just a general schematic of what a cell looks like with all the structures inside of it. So we have the overall cell, and then the structures inside are organelles because they're like miniature organs, right? So not all of these are going to be present in every cell, but most of them will be present. So we're going to go through what all of these are in the coming slides. The basic structure of a cell is that we have an outer membrane that's called the plasma membrane. And what that does is it separates the cell from its external environment. So otherwise, all the contents of the cell could just float away to who knows where, right? So that membrane is going to be the boundary of the cell. And so inside of the cell, we have the cytoplasm. And the cytoplasm contains everything inside the cell. So inside the cytoplasm, we have cytosol, which is the fluid that suspends all of the structures inside. The organelles, which are the structures inside the cytosol, which is also inside the cytoplasm. And those organelles are the miniature organs that will serve to perform different functions for the cell. And then probably the most important organelle is the nucleus. And the nucleus is the center of the cell that you see on the right. It's the biggest structure. And it's actually the only structure you can see microscopically at normal levels anyway. And then this nucleus is really important because what it does is it controls cellular activities and it houses the DNA for the cell. So basically all of the information is here. Now the cell is surrounded by interstitial fluid, which is also known as extracellular fluid. You can call it either, it makes no difference to me, but we'll refer to it as both throughout this lecture. And the interstitial fluid or extracellular fluid is mostly comprised of water. So moving into the lecture for today, we're going to proceed through the topics again in order of your textbook, for the most part, in order to help you stay organized so you can keep following along and take little breaks. So today's lecture is pretty long, so I think you'll be especially glad that we have this format today. So first, we're going to start off talking about the plasma membranes. The plasma membrane is comprised of 3 of the 4 organic molecules: lipids, proteins, and carbohydrates, excluding nucleic acids. So you'll notice that lipids are listed first, and that's because they're incredibly important because they are a major component of phospholipid molecules that we studied previously. And so the phospholipid molecules are those that create ultimately the majority of that structure of the plasma membrane. It creates the wall. So the lipid part is there because it comes from the lipid tails of the phospholipid molecule. Cholesterol is also another lipid that's involved in the wall, and that is there to help stabilize and stiffen the plasma membrane. Now, the proteins that are included in the plasma membrane are integral proteins and peripheral proteins, and they're exactly what you think. Integral is integrated into that membrane itself, whereas peripheral are proteins that are just hanging out at the periphery, so they have different responsibilities. So the integral proteins, you'll see by the way, has a little plus next to it, and that's to indicate that there are way more integral proteins than there are peripheral proteins, which makes sense when you think of what they are and what they're doing. So integral proteins include things like channels, carriers, receptors, anchoring proteins that help to anchor a cell in place, and enzymes that catabolize reactions. There are also peripheral proteins that are out, again, at the periphery of the cell, and so they're there for things like signaling to other cells and to create receptors. So recognition proteins are part of this, enzymes can be peripheral proteins, and another important point about peripheral proteins is that the receptors will bind to ligands. So receptors, when they bind with a ligand, will change the actual cellular activity of that cell. So the example that's given in the textbook is of insulin, which is a ligand that floats along throughout the extracellular fluid, and when that ligand binds with the receptor, what happens is insulin will then trigger a whole series of events, which is called a cascade. And this cascade of events is going to change the action of the cell in the way that it absorbs glucose. The third is carbohydrates. So carbohydrates are important because they form the glycocalyx, which is just really a number of large molecules that extend out beyond the outer surface of the membrane that create kind of an outer layer, if you will. So that glycocalyx is responsible for lubrication and protection. It also helps to anchor the cell and locomote it around if needed. It's involved in cell signaling, adhesion, binding, specificity, and also recognition. The plasma membrane is selectively permeable, which means that it basically functions like a bouncer at a bar. So it allows some passage and also denies other passage. So it regulates entry and exit of everything from ions through to complex molecules. It's also very important helping to maintain shape and isolation, but I think the most fascinating thing is that it's responsive to the changing conditions of the extracellular fluid that is surrounding the cell. So if, for example, the cell requires more sodium, the cell can upregulate the amount of sodium that can come in through the plasma membrane to meet its needs. And in so doing, it's able to maintain homeostasis for itself. So the plasma membrane being selectively permeable means that it's also semi-permeable, meaning that it's going to allow some things to enter and restrict access to others. So it's permeable or permissible. It allows passage of lipid-based substances, some small molecules, and non-polar molecules. Water molecules have a unique opportunity to pass through the wall, and they pass through something called an aquaporin. Aqua meaning water and porin meaning pore, right? So it passes through water pores. Now, other charged molecules are going to have to take other mechanisms that come through the plasma membrane, and these are more regulated. We'll talk about these coming up. This is figure 3-2 from your text, and what it is showing up close is a schematic of the phospholipid bilayer and the structures that are in the plasma membrane. So there's a lot going on here, but let's break it down step by step. So first off, what do we know? We know that the primary structure is the phospholipid bilayer. And so we see all of those little round red areas, and those are all parts of the hydrophilic end of the phospholipid molecule, right? So those little sticks that you see sticking down, those are the legs or the tails, which are the lipid ends of the phospholipid molecule. And so the lipid ends face each other, as you can see. So that's what creates the lipid barrier. And then the round molecules, the phosphate groups, are facing both out of the cell and into the cell, because both the exterior aspect of the cell is surrounded by extracellular fluid, which is watery, and the interior of the cell also has a watery fluid too. So we have the hydrophilic ends directly in contact with the watery outer aspects and inner aspects. You'll also see these little yellow kind of goobs that are stuck in between the phospholipids, and these are cholesterol. And so that's there to help stiffen and support the wall. We also have on the left, the largest structure that you see is a gated channel, and that's one mechanism of passing larger molecules through the plasma membrane. In the center, what we have is an integral protein with a channel. So remember, integral proteins are those that are integrated into the wall. So you can see that this channel here will allow passage directly through it for certain molecules, and it is an integrated, integral part of the membrane. There's also integral glycoproteins, which are also, again, integrated into the wall that you see over to the right. And then inside the cell, you're going to see the beginnings of the cytoskeleton, which is basically just the scaffolding or structure of the interior of the cell. So again, just for your notes, the phospholipids in the plasma membrane are there to comprise the majority of the phospholipid bilayer, and that includes hydrophilic heads, which are the phosphate group, and the hydrophobic or water-fearing tails that are formed by the nonpolar fatty acids. Those face each other, and those are composed of the lipids. So the lipid barrier has the two layers of the hydrophobic tails facing each other, and there's also an inner layer of the plasma membrane in between that too. And again, the phospholipid bilayer is going to allow some small, nonpolar, and fat-soluble substances to just diffuse freely right through the wall. They just walk through the wall like it doesn't even exist. And then there are water-soluble substances that are polar, and these can't move freely with the exception of water. And again, remember, water comes through the aquaporins. Cholesterol is an extremely important structure in the phospholipid bilayer cell membrane. And it is very important because it does a couple things. It stiffens and supports the wall, but that also increases fluidity and stability of the membrane. And so it's embedded in between the lipid tails that you can see in images that are present in your text. Figure 3-2 is probably the best one for being able to see the way this works. Channels are an integral membrane protein, and they're pretty straightforward. Basically, a channel is something that a boat could just go right through, and similarly, this will allow things like water and other hydrophilic solutes to just flow right through. So provided that the molecule is the right size and the right charge, it just passes on right through the plasma membrane without any interference. A carrier protein is a type of integral membrane protein in that you can see on the image on the right, the purple structure, which is the carrier protein, is integrated into the phospholipid bilayer of the plasma membrane. And so what the carrier protein does is it will transport only a very specific solute across the plasma membrane, either in or out. So the best example here I can give you is that of a boat going through a locks. So a sailboat might be the right size to be able to fit through a smaller locks, no problem. But a 1,000-foot ocean liner, for example, may be too large for the gates to close on both sides of the locks to chaperone them through. So in this case, you can see that there are going to be very specific types of proteins or molecules that can be transported across by the carrier protein, but others are going to be too large to be transported through. Receptor proteins are a little bit different. Now, they're also an integral membrane protein in that they are integrated into the plasma membrane itself, but what they do is they bind and respond to ligands, like I discussed previously with insulin. So a ligand could be anything ranging from an ion all the way up to a complex hormone. So whatever the molecule or ion is, the receptor proteins bind to that ligand, and then what happens is there's a chemical cascade that happens inside the cell as a result of this binding, and it will change the entire cellular activity of the cell. So insulin, again, as the example, was a ligand that once bound to a receptor protein will change the cellular activity, adjusting the rate of glucose absorption by the cell. Other membrane proteins include structural attachment proteins, and these are effectively stabilizers for the structure of the plasma membrane and the cell. So they'll attach to either the inside or the outside structures of the cell. Other membrane proteins can also include enzymes, and these are proteins in some cases, and they will function to speed up or facilitate or catalyze a chemical reaction. Let's talk about membrane carbohydrates now. Probably two of the most important are going to be glycoproteins and glycolipids. So if you look at the image on the right, which is figure 3-2 in your text, you'll see that these purple branching structures sticking out from the exterior surface of the cell membrane into the extracellular fluid are glycolipids of the glycocalyx, right? And these sticky glycocalyx branchings are really important because if they're sticky, they allow the cell to stick to other cells, so it allows for cell adhesion. They also act as flags that help to mark the exterior of the surface of the cell for a number of things, and this is an important role in immune recognition. So each cell is going to have its own unique signature of carbohydrates on the cell surface, and it's also unique to each individual cell type and to the individual. Now, another important point about membrane carbohydrates is we know that carbohydrates attract water. And why is this important? Well, I mean, think of this in terms of dieting. If you cut carbohydrates from your diet, you're going to probably lose some weight pretty quickly. But really what you're losing is water because carbs attract so much water. So it's a really nice feature of the cell that we have these glycolipids of the glycocalyx sticking out because they attract water towards the cell's exterior surface, which allows for the cell to maintain homeostasis. Pop quiz question. Which of the following components is not a part of the plasma membrane? A, phospholipids, B, proteins, C, nucleic acids, or D, carbohydrates? Well, let's walk through this. We know that the plasma membrane is composed of a phospholipid bilayer, so we can rule out A. We know that proteins obviously play an important part in the cell membrane, so we can rule that out as well. Carbohydrates we just discussed about last, and that's things like glycoproteins and glycolipids, and they have a whole lot of important roles. And so the one thing we can rule out here for certain is nucleic acids. They are absolutely not a part of the plasma membrane. Which of the following is not a function of the plasma membrane? A, that it produces energy for cell use. B, that it encloses and supports the cell. C, that it controls the movement of materials into and out of the cell. Or D, aids in communication between cells. Well, we know, let's start from the bottom up. The cell is absolutely in communication with other cells because of the outer surface of the cell, which is the plasma membrane, right? So we can rule out D right away. C, controlling the movement of materials into and out of the cell. Well, that is the full purpose of the plasma membrane. I mean, that's absolutely imperative to know this. It also encloses the cell content, so that way the organelles don't just float out freely into the extracellular fluid. So the answer here is A, produces energy for cell use. Which component of the cell membrane is primarily responsible for forming a physical barrier between the cell's internal and external environments? Either A, the phospholipid bilayer, B, the glycocalyx, C, peripheral proteins, or D, proteoglycans. Well, D, proteoglycans are very important, and C, peripheral proteins are also important, but they are on the periphery. The glycocalyx only sticks out in certain branching areas, as you remember from that image that we just looked at, which is figure 3-2. And so the answer is A, the phospholipid bilayer is responsible for primarily forming a barrier between the cell's internal and external environments. Passage of a very specific hydrophilic solute from the extracellular fluid into the cell occurs through A, the plasma membrane, B, channels, C, carriers, or D, aquaporins. Well, aquaporins allow water to pass through, so that doesn't qualify. Carriers will carry specific items, specifically solutes, across the cellular membrane. Channels allow passage of things more freely, and the plasma membrane itself, of course, is the barrier to the cell. So the answer here is going to be C, carriers, because carriers allow for specific hydrophilic solutes to pass through. Just when you think the plasma membrane is kind of an abstract component of a cell, it is incredibly important in cancer diagnosis and in treatment. So this is not testable material, but I put this here for your interest. So there's a video that you can watch. You can select the link that I've included below. And what it is going to describe to you the way that HER2 signaling is able to be detected, and therefore if the HER2 receptor is present on a specific type of cancer cell, then that means that the cell will respond to Herceptin, which is a very specific type of chemotherapy. So this is the future of chemotherapeutic drugs. If we're able to detect that a cell is able to produce a very specific receptor, then we know that we can target that type of receptor with certain medications. So please take a moment to watch this. I think it's fascinating, and I think you're going to really enjoy it. There are some structures that are best to discuss now because they're membrane and cytoplasmic structures. So first, let's discuss cilia. Cilia are these very short, thread-like moving extensions that will stick up off the plasma membrane And what they do is wave in order to move fluid. And they wave sequentially in order to produce a mass effect together. Cilia are present in a couple different places in the body, and probably the two most common places you expect to find them are lining the trachea, which is our airway in between our mouth and our lungs, and also in uterine tubes. And the uterine tubes are going to be the structure that is in between the ovary and the uterus. So first let's talk about the trachea. So the trachea has cilia that help to propel things upward. In this case, it's not going to be propelling fluid necessarily, but anything in fluid. So let's say if you walk into a dusty room and you start to cough, as you cough, which is a big muscular reaction, you also have cilia that will help to wave whatever it is you inhaled in up and out of your body. Similarly, in the uterine tube, it's responsible for taking the ovum from the ovary to the uterus. And in the uterine tube, there is no muscular peristaltic contraction that would propel the ovum forwards, right? So in this case, we need something to move it along. So what we have are cilia here, which will wave in order to push the ovum along into the uterus. They do this by using two different types of stroke. One is called a power stroke and the other is the return stroke. So the power stroke is just like when you're swimming, doing the front crawl. The power stroke generates all the power to propel you forwards, whereas the return stroke is when you would drag your arm back just over the surface of the water, just sort of nicking it, not creating any additional force. So that is basic ciliary movement. Side note of clinical correlation. My husband's grandmother actually had Cartaginer syndrome, which is a ciliary dysfunction disorder. And so what happens in this case is the cilia in your body don't work. And when you have that, you have a number of different problems. So we just talked about how cilia line the trachea and uterine tubes. So if you don't have cilia in your uterine tube waving the ovum along, it's possible to have problems with fertility. Further, if you have ciliary dysfunction in your trachea, you may be subject to a lot of different infections in your lungs because you don't have the ability to expel infectious organisms in the same way that you and I might. A flagellum, which is singular, or flagella, which is plural, are really just long-moving extensions that stick off of the end of the plasma membrane. And flagellate is a verb that basically means whip, right? So the flagella whips along behind and it propels the cell forward. And there's only one cell in the human body that has a flagellum, and that is a sperm cell. And it's important to note that if there is any dysfunction whatsoever in the flagella of the sperm cells, that those cells would then have problems with fertility. Microvilli are also membrane and cytoplasmic structures that are kind of similar to cilia and flagella, but they're not in another way in that they don't move at all. They just stay put, and their primary reason for existence is to increase surface area. And this is really important. So as food is going through our digestive tract, the increased surface area allows for a greater time with contact with that food product. So that means that we can absorb both nutrients and water from the food better. So there are certain types of diseases and syndromes that will result in having blunted villi in the intestinal tract. And as a result of that, the patients will end up with a number of different absorptive issues and dysfunctions. All right, let's talk about organelles, the miniature organs that are present within the cell itself. Cytoplasm is everything inside the plasma membrane inside the cell. And this can be broken down to two main categories. The cytosol, which is just the fluid that allows the organelles to float along in. So the cytosol is fluid that has dissolved molecules and ions in it, and the organelles are all of the miniature structures that you see in figure 3-1 below that show you that there are things like the mitochondria and lysosomes, the nucleus. Those are all organelles that are specialized structures among the cytosol in the cytoplasm. The cytosol composition is incredibly important because it helps maintain homeostasis of the cell. So there's a higher potassium in cytosol than in the extracellular fluid, but a lower sodium in the cytosol than the extracellular fluid. Also, the suspended protein concentrations are higher in the cell than in the fluid surround it, which makes sense, right? Because the extracellular fluid is more watery. And the nutrient concentrations are actually lower in the cell than in the extracellular fluid. So that means the cell is holding on to all these nutrients like carbohydrates and lipids, amino acids, and the extracellular fluid is much more dilute. Organelles are broken down into categories of being either membranous or non-membranous. And what this really means is either they are in direct contact with the membrane of the cell, or they are completely individual entities floating along on their own. So the non-membranous organelles are ribosomes, the cytoskeleton, cilia, flagella, microvilli, and centrioles. And not all cells are going to have flagella and cilia and microvilli, for example, but those are non-membranous organelles. By contrast, membranous organelles include the endoplasmic reticula, which is reticulum as a singular, and that is both smooth and rough endoplasmic reticula, the Golgi apparatus, lysosomes, peroxisomes, mitochondria, and the nucleus. Organelles can be broken down into two categories, those that are non-membranous and those that are membranous. And when we're talking about membranous, what we're talking about is having a definitive boundary. So the non-membranous organelles do not have a definitive boundary, whereas the membranous organelles are surrounded by a membrane and they are entirely enclosed units on their own. So the non-membranous organelles will include things like ribosomes, the cytoskeleton, cilia, flagella, microvilli, and centrioles. By the way, it's important to note that not all cells are going to have cilia and flagella and microvilli, etc., but those are all non-membranous organelles in general. The membranous organelles will include endoplasmic reticula, or reticulum, which is singular, and this includes both the smooth and the rough endoplasmic reticula, the Golgi apparatus, lysosomes, peroxisomes, mitochondria, and the nucleus. The cytoskeleton is a non-membranous organelle, which is the protein scaffolding of the cell. So it gives the structure to the cell overall. And it's responsible for movement. And so there are the following things listed that are highlighted here for you that we're going to discuss in more detail coming up in subsequent slides. But first, know that microfilaments, and actin is an example of one, is going to be responsible for helping move the cell, whereas intermediate filaments that are a little bit larger are going to be more structural in nature. And there are also microtubules, which are going to be important for transport and movement of chromosomes, whereas finally, thick filaments, which are the largest, are going to be both important for movement and structural. All I want you to hone in on this image here is if you look to the primary image on the left, you're going to notice that there are three different types of subunits that are described. So starting at the very bottom, we have actin filaments, which are the thinnest and finest, and you see the arrangement of them and how it's different than the two other structures, the intermediate filaments and the microtubules. So the microfilaments, or actin filaments, they are the thinnest, and they're about 8 nanometers in diameter. Moving up one step, moving to the right, you'll see intermediate filaments, and they're exactly what you think. They're intermediate size. And so the intermediate filaments are about 10 nanometers in diameter. And then we move up into microtubules, which form tubes, right? They're microtubes. And so these are going to be about 25 nanometers in diameter, but the walls themselves are only about 5 nanometers across. So that leaves an area on the inside of about 50 nanometers of space. And if you look to the right, you're going to see a scanning electron micrograph, which is what the SEM stands for. This zoomed in at a 60,000 times view, and it is showing you what an intermediate filament looks like as opposed to a microtubule. So this slide just compares and contrasts intermediate filaments from microfilaments. So the intermediate filaments are larger, of course, than the microfilaments, and so they're there to provide some structural support. They also help to anchor organelles to specific locations inside the cell, so they're not just all bouncing around, hitting each other and becoming damaged. So the intermediate filaments, again, are protein fibers. They're about 10 nanometers in diameter. The microfilaments are very thin. In fact, they're really, really delicate. So they're only about 8 nanometers in diameter. And what they do is they'll interact with a number of different things. And the example you see below is an actin filament. And in skeletal muscle, actin and myosin are going to work together to create movement and muscular contraction. Microfilaments are also important because they affect the viscosity of the cytoplasm, and again, these also act as an anchor for membrane proteins. Microtubules are another non-membranous organelle in the cytoskeleton, and the microtubules form the main portion of the cytoskeleton. So these protein tubules are built from a protein called tubulin. And so you can see the tubulin subunits on the right that are each 5 nanometers in diameter, but overall the structure is 25 nanometers in diameter. So these microtubules are really important because they do a number of things. So they give the cell its shape overall, and because they can change the shape of the cell, that can help with cell movement. So there's a lot of responsibility for these little tiny microtubules. But during cell division, they also have a different role. So what they do is that they'll basically distribute these chromosomes to different ends of the cell, and they create a network of microtubules called the spindle apparatus. We'll talk more about this later, but for now, just know that the microtubules have a lot of different roles and are incredibly important as the main portion of the cytoskeleton. The centrosome is a non-membranous organelle that's located right next to the nucleus in the cell. And the centrosome is the organization center for cytoskeletal microtubules. So let's look at the image on the right. In color, what you're going to see are 9 sets of 3 microtubules, all arranged circumferentially around the center. And you're going to notice that each of those sets of 3 is set at a right angle or perpendicular to the immediately adjacent microtubule set. So the 9+0 microtubule array that we describe this as is literally just explaining that there are 9 sets of centrioles and that there's nothing in the center. So the 0 indicates that there are no microtubules in the center. The centrioles are incredibly important because they're required for cell division. And primarily what they do during cell division is that they're going to help form the spindle apparatus, which helps to organize and pull chromosomes so that way mitosis can occur. So if a cell does not have a centriole, the take home here is that it cannot further divide. So things like red blood cells, skeletal muscle, cardiac muscle, and most neurons are all going to fit into this category of cells that have no centrioles and therefore cannot further divide. Also, centrioles are responsible for forming the basal bodies, which form as the base for some cellular extensions. I hope you recall our discussion about cilia and flagella from a few slides ago, because these are also non-membranous organelles. Cilia can be divided into the categories of either being non-motile or motile, which really just means do they create movement. So non-motile cilia do not create movement. They have a totally different responsibility. Non-motile cilia are going to act as a sensor to the environment. And this is important because each cell that has this non-motile cilia will only have a single cilium. So that one single primary cilium is going to be responsible for coordinating embryonic development and maintaining homeostasis at the tissue level. So because that cilium is going to be sticking up off of the plasma membrane, it's going to be able to interact with the extracellular environment. So the non-motile cilia have a 9+0 area arrangement, so they look just like centrioles, like what we talked about before. Now contrast that with motile cilia, which do create movement. These are the guys that we talked about earlier that are present in the respiratory and reproductive tract that wave along bacteria or some sort of dust out of your lungs and your trachea. And also the reproductive tract cells are going to help have cilia wave and ovum along through the uterine tube. The motile cilia have a different organization. They have a 9+2 array. So what that's meaning is in the center of the array, we have 9 of those sets of triplets around in the centrioles and in the non-motile cilia. But in the motile cilia, we still have the 9 all the way around, but we also have 2 in the center. So it's just a slightly different configuration. So motile cilia are anchored to a basal body beneath the surface. And remember, we just talked about this a moment ago when we talked about centrioles. Centrioles will also form that basal body, which is basically what anchors the motile cilia to the plasma membrane. So next we have flagella, plural, or flagellum singular. And again, these are only present in sperm cells, so therefore there's only one flagellum per cell, per sperm cell. And these also have a 9+2 array, like the motile cilia. Ribosomes are another non-membranous organelle, and these are responsible for protein synthesis. The number of ribosomes that you find in a cell will be proportional to the type of cell that it is and how much protein synthesis it needs to actually manufacture. So for example, liver cells or hepatic cells, they're responsible for manufacturing blood proteins. So these cells are going to have many more ribosomes than, say, fat cells, which really only synthesize lipids and don't have a major role in protein synthesis. So Proteins can be formed as a large and a small subunit in the nucleoli and as they come together. So before protein synthesis actually begins, that small and the large subunit have to come together and they join with a strand of messenger RNA. And that's another type of RNA. But anyway, that's the point at which protein synthesis can actually begin. So you will see ribosomes in two different locations, either freely floating throughout the cytoplasm or fixed to the endoplasmic reticulum. So the freely floating ribosomes are going to have a responsibility of synthesizing proteins that can be immediately delivered into the cytosol or the cytoplasmic fluid. On the other hand, the fixed ribosomes have a different responsibility. They're going to synthesize proteins that have a totally different destination or endpoint. So instead of just releasing their product into the cytosol, these are products that need to be excreted outside of the cell or potentially to a different location within the cell. So these subunits are formed from proteins and ribosomal DNA. Our last non-membranous organelle are proteasomes. So proteasomes are responsible for breaking down protein. So they contain proteolytic enzymes or proteases. So remember, anything that ends in ACE, ASE, is going to be an enzyme. And the name proteolytic tells you proteo is protein and lytic, lysis, right? So we're breaking down protein. So the role of proteasomes is to remove any of these damaged or denatured proteins that are found in the cytoplasm. And this also includes virus-infected cells. So you can see how this has an important role in our immune response. So these cytoplasmic enzymes are going to attach to ubiquitin, which you can remember from the word ubiquitous, meaning that something is pretty much present everywhere. So they attach to ubiquitin, which is a tag for proteins that need to be recycled. So then it can be moved to the proteasomes, at which point the proteasome can break down the protein into amino acids and small peptides, so that way they can be safely released into the cytoplasm and not damage any part of the cell any further. Our first membranous organelle is the endoplasmic reticulum. First, let's talk about what a membranous organelle is. So a membranous organelle is going to be one that has its own distinct membrane that is separate from the surrounding cytosol or cytoplasm. So in this case, let's talk about the endoplasmic reticulum. You can see that it is right up against the nucleus because it is having its network right up with the nuclear membrane. In fact, it's not just up against it, it's continuous with it. So this is really important because endoplasmic reticulum is a very important part of the cell that has a lot of responsibility. So a reticulum is just a network, an endo meaning within, and plasm being cytoplasm. So within the cytoplasm, we have a network, and that's what the endoplasmic reticulum is. And this network is responsible for storage, and so it can store a variety of different types of molecules or other materials that are absorbed into the cytosol from the extracellular fluid. It can also detoxify a number of drugs and toxins and then neutralize them with enzymes so that way they can't damage the tissue or the cell any further. The endoplasmic reticulum is also involved in transport, so various molecules can travel from place to place within the endoplasmic reticulum itself. And then lastly, it's responsible for protein synthesis, but it also will synthesize lipids and carbohydrates. So you'll find endoplasmic reticulum in one of two different layouts. One will be called rough endoplasmic reticulum, and the other one is smooth endoplasmic reticulum. So the rough endoplasmic reticulum is going to have ribosomes on it, and the ribosomes, by definition, are what make it rough endoplasmic reticulum. So you'll see in the image in the middle that there's a piece of endoplasmic reticulum that looks very much like coral if you're a scuba diver. And on the left, you'll see that there are ribosomes studding the whole entire exterior surface of the endoplasmic reticulum. On the right, you're going to see that the endoplasmic reticulum is very smooth and has an absence of ribosomes. So it's smooth if it is without ribosomes, and in rough if it does have ribosomes. And if you move a little bit further over to the right yet, you're going to see a fantastic image of a transmission electron microscopy zooming in on the endoplasmic reticulum at 111,000 power. And what we're looking at here is you can see there's some like horizontal looking lines there that look like tubes, and that is the rough endoplasmic reticulum with fixed attached ribosomes on it. And then also there are some free ribosomes kind of floating freely out in the cytosol. And you can also see down at the bottom, if you follow the lines, some smooth endoplasmic reticulum as well. The smooth endoplasmic reticulum is primarily responsible for lipid synthesis, so it synthesizes things like cholesterol, steroid hormones, phospholipids, and triglycerides. And so because you see steroid hormones there, you can presume that if it's synthesizing lipids, that you should expect to see a greater proportion of smooth endoplasmic reticulum in cells of the reproductive tract that are going to synthesize steroid hormones. It's also responsible for glycogen synthesis and storage, and glycogen is an incredibly important form of energy for us. So that gets stored in two places primarily, in our skeletal muscle, so that way we have access to quick energy, and also in our liver cells as well. The smooth endoplasmic reticulum is also responsible for detoxifying and inactivating drugs, so we talked about that a little bit earlier, and it has the same effect as the rough endoplasmic reticulum in that the vesicles that are formed are bound for the Golgi apparatus, which we'll talk about coming up shortly. So you can expect to see a high proportion of smooth endoplasmic reticulum in any cell that is responsible for a lot of lipid synthesis. The rough endoplasmic reticulum is more involved in protein synthesis and packaging, and for this reason it gets referred to as a UPS store. It's basically a combination of a workshop and a shipping warehouse. So as a workshop, it's going to synthesize protein, and as a shipping warehouse, it's going to package that protein and then ship it out. So it's shipping it out in vesicles that are bound for the Golgi apparatus. You can expect to find a higher proportion of rough endoplasmic reticulum in any cell that's going to be manufacturing digestive enzymes. So for example, in the pancreas, you're going to expect to see a lot of rough endoplasmic reticulum. I really love this schematic because I think it illustrates well exactly what happens in the rough endoplasmic reticulum. So you can see on the left that there is a ribosome attached to the rough endoplasmic reticulum, and in that ribosome we have some newly synthesized proteins. So those new proteins get chemically modified in the endoplasmic reticulum, and then those proteins are going to move along because the endoplasmic reticulum is a reticulum, right? It's a network. So the protein moves along through the network until it gets to a specific point at which the rough endoplasmic reticulum actually pinches off a part of itself and creates a transport vesicle. This is almost like something out of Star Trek. So it pinches off this transport vesicle, and once it buds off, it pinches off completely, and then it can float out to go to the Golgi apparatus, or it could also be secreted to the plasma membrane to be excreted out of the cell entirely. The Golgi apparatus is another organelle that is membranous, and it looks somewhat similar to the endoplasmic reticulum, but in my mind, I think of the endoplasmic reticulum as looking more like a piece of staghorn coral, whereas I think that the Golgi apparatus looks like a stack of dinner plates that maybe Salvador Dali drew, like the persistence of memory, because everything is kind of drooping in a bizarre way. But either way, the Golgi apparatus and the endoplasmic reticulum both have cisternae, which are really just these water-holding flattened sheets. So this is primarily composed of the cisternae, whereas the endoplasmic reticulum will have cisternae, but it's also going to form hollow tubes and various other shapes as well. So the Golgi apparatus is responsible for modifying and packaging proteins that include things like hormones and enzymes and various membrane components as well. Figure 3-7 in your text is really excellent at showing protein synthesis, processing, and packaging from start to finish. So let's walk through this in sequential order. So let's start off in the nucleus at step 1. So protein synthesis is going to start when a gene on DNA is going to produce messenger RNA. So the messenger RNA is going to leave the nucleus and go to a ribosome that is either free in the cytosol or fixed to the rough endoplasmic reticulum. If the mRNA goes to the free ribosome, the protein synthesis occurs there, and the protein is going to be released into the local cytosol. If, on the other hand, the mRNA goes to a rough endoplasmic reticulum ribosome, then that is the site of the protein synthesis, and the newly synthesized protein is going to be shipped throughout the network of the endoplasmic reticulum until it gets to a point that can be pinched off and creating a transport vesicle. So step 4 is pinching off, and then you can see as we move towards step 5, these transport vesicles are carrying these products over towards the cis face, or the receiving side, of the Golgi apparatus. So there are going to be several or even multiple of these transport vesicles all arriving at the same time. So when this happens, there will be yet another protein and glycoprotein modification. So after these chemical reactions have occurred, then we'll have newly packaged products that are ready to be shipped out. So they'll be shipped out on the transface or the shipping side of the Golgi apparatus, which is also closer to the plasma membrane of the cell. So the three different things that can come from this are going to be either a lysosome, a secretory vesicle, or a membrane renewal vesicle. So the lysosome is going to be important for having digestive enzymes to help clean up the cell, or the secretory vesicle will help fuse with the plasma membrane, and then once that happens, it can expel its product into the extracellular fluid by a process called exocytosis, which we'll discuss later. The third thing that can happen is we can have a membrane renewal vesicle. And so this is going to add new lipids and new proteins to the plasma membrane to continually renew it and help it maintain its integrity. Lysosomes are a membranous organelle that I think of as the dirty job of the cell. So lysosomes contain digestive enzymes, and that's important because of what they do. They clean up the cell. So they clean up things like cellular debris, bacteria, even old organelles that may be damaged, and nutrients that are taken in through endocytosis, and they will also turn polymers into monomers. I told you this course builds upon itself. So lysosomes really just create an isolated environment for risky chemical reactions that could potentially damage the cell. So there are two different types of lysosomes. There are primary lysosomes that contain inactivated enzymes, and then secondary lysosomes are only formed when that primary lysosome is going to meet with a piece of a damaged organelle. And once that happens, it becomes activated and therefore can start digesting its contents. Lysosomes are yet another membranous organelle, and I like to think of lysosomes as basically being like the dirty job of the cell. These contain the digestive enzymes that help to clean up anything in the cell that's undesirable. So that could be anything ranging from cellular debris, bacteria, an old organelle that could be dysfunctioning, nutrients that can be taken in through endocytosis, and also it'll turn polymers into monomers. I told you this course builds upon itself. So really what lysosomes do is they provide an isolated environment for chemical reactions that could potentially damage the cell to occur safely. So there are two different types of lysosomes. There are primary lysosomes, and these contain inactivated enzymes. The secondary lysosomes are the ones that actually do the action, but they are only activated when they run into a potentially damaged organelle. So once that happens, then they can get to work. Now, if the lysosome expels its contents into the cell, those digestive enzymes en masse can destroy the cell in a process called autolysis. So autolysis is really just when the intrinsic enzymes of the cell kill it from the inside out. This is different than something like putrefaction, which would be when bacteria from the outside in would destroy tissue. Figure 3-8 in your text shows you how the lysosomes actually work from start to finish. So starting at the top, the Golgi apparatus will release a primary lysosome, and that primary lysosome can go one of three different directions. So one could be, it could come into contact with a damaged organelle, like you see on the left. In this case, it's damaged mitochondrion. And so when that happens, it becomes activated as a secondary lysosome. Some contents of the secondary lysosome can be reabsorbed into the cell, but much of it will be expelled out of the cell by a process called exocytosis. The second thing that could happen is that primary lysosome from the Golgi apparatus could also become a secondary lysosome because it comes into contact with something brought in from the extracellular fluid in a process called endocytosis. So that extracellular solid or fluid will come into a vesicle called an endosome. That endosome will fuse with a primary lysosome, creating a secondary lysosome, and then the product of that secondary lysosome can also be potentially reabsorbed back into the cell or expelled from the cell into the extracellular fluid in a process called exocytosis. The third thing that could happen is that primary lysosome from the Golgi apparatus could also totally break down. And this happens over time if there is any damage to the lysosome itself. And so eventually, this lysosome will release its digestive enzymes into the cytosol. And when that happens, those digestive enzymes are so harsh that it will essentially demolish the entire cell in a process called autolysis. Peroxisomes are similar to lysosomes, but they're different in a number of ways. So first off, they're smaller and they carry a different group of enzymes than the lysosomes do. So they break down different things. So peroxisomes are responsible for breaking down fatty acids and other organic molecules. But when that happens, that breakdown results in some hydrogen peroxide being generated, and that hydrogen peroxide can be very damaging to the tissue. So basically what has to happen is that hydrogen peroxide has to be broken down into something safer, right? So catalase, which is an enzyme, anytime you see ase at the end of something, it's an enzyme. Catalase is going to break down hydrogen peroxide into two very safe things. First is water, and the second is oxygen. So these are incredibly important cells, as you can imagine. You can see peroxisomes present in pretty much all cells, but you're going to see them more frequently in a metabolically active cell. So liver cells have an awful lot of peroxisomes. But I think the neatest thing about peroxisomes is going back to my very first point, which is that they're produced by existing peroxisomes. This is almost like a which came first, the chicken or the egg sort of thing. So I hope that helps you to be able to remember that peroxisomes are a little different than lysosomes. Pop quiz. The mitochondria is the what of the cell? I'm certain that probably every single one of you listening is remembering it's the powerhouse of the cell from elementary school, because that was drilled into my mind, and I think it was probably drilled into yours too. So what does that mean if it's the powerhouse of the cell? Well, it means that it produces the energy that the cell needs in order to do all of these various activities. So the energy that it produces is in the form of adenosine triphosphate, or ATP. Also, mitochondria have their own DNA, and this is abbreviated as mtDNA for mitochondrial DNA. Just make a note not to confuse this with messenger RNA, which is very different. So let's look at the structure of the mitochondria. So first, it has a double membrane, which is unusual. Most, if not all, other membranous organelles only have a single membrane. So this double membrane is set up so that way the outer membrane is what you would think of as a normal membrane. It encapsulates the whole entire organelle. But the inner membrane folds in to create cristae, which you see in the images down below in the bottom right. So the cristae are those long, almost finger-like extensions similar to microvilli, extending into the center, which is called the matrix because it's filled with fluid. So the cristae serve the purpose of increasing the surface area in the cell, just like the same way that microvilli do. The image below is a meme that went viral a while ago among science nerds. And so mitochondrial DNA is something that's been understood to be given to us only by our mothers. So that's an image of a mitochondrion below. And this is actually something I sent to my mom on Mother's Day a little while ago. If you're interested in some supplemental reading that is not testable material, the link above that I've included from the journal Nature indicates that there's some recent research showing that in some rare instances, fathers can actually provide some mitochondrial DNA to their offspring as well, which is groundbreaking news. Anyway, if you haven't read anything through Nature before, this is effectively the journal. So if you ever have groundbreaking, major scientific breakthroughs, they're going to be published in Nature. So this is just something to read for fun if you're interested. Mitochondria create energy as ATP, or adenosine triphosphate, which is the energy currency of the cell. And it does it by a process called glycolysis. Figure 3-9 in your text illustrates well exactly how this works. In general, glycolysis is breaking down glucose. So it breaks down glucose into pyruvic acid, and then pyruvic acid is transformed into three things: carbon dioxide, ATP, and water. So because oxygen is involved in this process is known as aerobic metabolism. This will be a concept that comes back to you when you take pharmacology, because there are certain microorganisms that use aerobic metabolism and others that use anaerobic metabolism, meaning not using oxygen. This will impact what types of drugs are given as an antimicrobial therapy, for example, antibiotics. Let's move into section 3-3 and discuss the nucleus. The nucleus is the brain of the cell, so it controls all cellular activity and replication. So the nucleus is a very important structure, And let's talk about that structure first. So it has a nuclear envelope, which is a double membrane, similar to the mitochondria. And in that double membrane, there are pores that allow different molecules to pass through, and that would be called the nuclear pores. Makes sense. So inside the nucleus, there's going to be the nucleoplasm, which contains matrix, which is really just support filaments that help to provide structure of the overall organelle. There's also a transient organelle, meaning that they're only visible and present at certain times, and that's called the nucleoli, or singular nucleolus. So the nucleolus you can see in the bottom right image, which is figure 3-10, and it's that dark center area of the nucleus. So the nucleoli are made of RNA, enzymes, and histones, and they're responsible for synthesizing ribosomal RNA and then assembling ribosomal subunits. A nucleosome is when DNA coils around the histones, and that's important because it allows a lot of information to be compacted into a small space. So the genetic material that's present in the nucleus takes one of two different forms, contingent upon whether it's dividing or not dividing. So if a cell is not dividing, that DNA is going to be just loosely coiled into something called chromatin. So it's just kind of a tangle. But then when the cell is getting ready to divide, then those chromosomes are going to become much more organized and tightly coiled. And so that's when you would see the chromosomes that we know as part of a normal karyotype. Nucleic acids are really what has all of the information in them in the nucleus. So DNA is deoxyribonucleic acid and RNA is ribonucleic acid. And you should be able to spell both of those very easily. So make sure you study that. The genetic code is broken down as follows. So the genetic code is really what the chemical language of DNA is, okay? So it is a sequence of nitrogenous bases, and the nitrogenous bases are adenine, thymine, cytosine, and guanine. And those get abbreviated to the first letter of each of those names. So it's A, T, C, and G. So triplet code is when you take 3 of those nitrogenous bases and 3 of them together are going to create one amino acid. So for example, TGT, which is thymine, guanine, and thymine again, creates the amino acid cysteine. So a nucleotide is when you take a nitrogenous base and a sugar, which is either oxyribose or deoxyribose, contingent on whether we're discussing RNA or DNA, and a phosphate. And a gene is going to be the DNA instructions for one entire protein comprised of those triplets. And this is the functional unit of heredity. The image that you see on the right is photo 51, and this is famous because it was taken by researcher Rosalind Franklin, who was largely an underdog and unheard of, but she has a very important role in the discovery of DNA. So Watson and Crick are of course credited with discovering the spiral helix structure of DNA, but it wasn't until they went down the hall and saw her notes. So Rosalind Franklin had taken all these excellent images of X-ray crystallography, and she was able to take this picture here, which you see this X. And so this is a 2-dimensional image, which once Watson and Crick saw this, they were able to deduce that there is in fact a spiral helix shape to DNA. Now we'll discuss how DNA controls the cells. Figure 311 in your text shows you how DNA is organized in the nucleus when the cell is either prepared for division or it is not prepared for division. So first off, let's look at the bottom. You can see the standard DNA double helix structure, and there are nucleosomes, which are when that region wraps around the histones. So you can see that there's a lot of information that can be trapped in this area because it's so tightly coiled. So now we have 2 different cells to look at the top left of the image. One is a cell with a nucleus that's prepared for division, and the other one below it is a non-dividing cell. So the cell that's prepared for division has organized genetic material inside the nucleus in the form of chromosomes. So these take the shape of the letter X, and we're able to see these in a normal karyotype. Now, if you look down below at the lower cell that's not dividing, when we zoom in to look at what's happening in the nucleus, you'll see that the chromatin is diffuse. It's just totally tangled around in the nucleus, hanging out, relaxing. It's not going to work. It's just totally taking it easy on the weekend, right? So the chromatin is totally in a different configuration whether the cell is dividing or not dividing. Some of the structures you're probably familiar with when you look at chromosomes are chromatids, which are condensed DNA in cells that are prepared for division. So you can see at the very top center of the image, there are two sister chromatids present. And this is illustrated in a more simple image down below at the bottom left. So you can also see that there's a yellow focus in the center of those two sister chromatids that join them together, and that's known as the centromere. Also, by the way, you can see the letters P and Q, and this is how we differentiate which arm of the chromatid we're discussing. So the P is known as the short arm and the Q is the long arm. And P in French, by the way, is short for petite or little. So that's how I've always remembered that P is the short one. So here are some definitions just before we go on. So a triplet is going to be 3 nucleotides found on the DNA that specifies 1 amino acid. Now, a codon is similar. It's 3 nucleotides, but it's found on the messenger RNA that codes for 1 amino acid. An anticodon is 3 nucleotides found on the tRNA that are complementary to the codon. Now, RNA is set up as a single strand, and it's important because it allows for information transfer from the DNA into the cytoplasm. And it has enzymatic properties, which of course will help facilitate a reaction. And instead of being the ATCG nucleotides that we discussed in DNA, we have one change in RNA, and that is that the T changes from thymine to uracil. So it's AUCG. So what is the role of ribonucleic acid, or RNA? Well, there are a few different types, so let's discuss them. So mRNA, or messenger RNA, is responsible for carrying a copy of the gene from the nucleus to the cytoplasm for protein synthesis. Now, ribosomal RNA is going to be important because it forms part of the ribosome, just like the RNA tips you off. And then we have tRNA, which is transfer RNA. And so it's going to carry amino acids to the ribosome also for protein synthesis. Before we get into transcription and translation in greater detail, let's just take a 30,000-foot view of protein synthesis in the cell. So first off, we know that the nucleus houses the DNA, which is all of the information necessary to produce the proteins. But DNA is too big to leave the nuclear envelope via the nuclear pores, so we need to have an additional mechanism. This is where messenger RNA comes in. So transcription is when one DNA strand is going to result in messenger RNA, so that way it can leave the nucleus. So messenger RNA is a complementary copy to all of the information that's in that DNA strand on its own. So the messenger RNA will leave the nucleus, and it's going to go to a ribosome, which is the site of protein synthesis. Amino acids, which are the building blocks of proteins, are going to be carried to the ribosome by transfer RNA. And then finally, in the process of translation, all of the information that was present in the messenger RNA is going to be rearranged into amino acid to create a polypeptide chain. So here's another 30,000-foot view of protein synthesis. So let's describe the differences between transcription and translation. So transcription is DNA going into RNA, okay? So in this case, we're going to have 3 DNA nucleotides, and they will become 3 RNA nucleotides. And this all occurs in the nucleus. Translation, on the other hand, is a process that occurs in the cytoplasm. So this is when RNA is going to become a protein that can be used by the cell. So 3 nucleotides will become 1 amino acid in the process of translation. Let's discuss protein synthesis verbally and in text form before we look at it visually. So let's discuss transcription. Gene activation is the very first thing that has to happen before transcription can begin. So gene activation begins by histones being temporarily removed. And then the first step will be RNA polymerase binding. And what this is basically the enzyme RNA polymerase binding to the promoter of a gene on the template strand. The second step is when RNA polymerase links nucleotides. So what will happen is this is like a zipper formation. So it starts at a start signal in a promoter region, and as it goes down the strand, it's going to read the DNA code. So after that, it's going to bind nucleotides together to form messenger RNA in a 3-base sequence known as a codon. The third step is when mRNA detaches. So the enzyme and mRNA strand are going to detach from the DNA at the stop signal, which is another codon. And then finally, this information has to be processed. So RNA processing is a really important step because at the end of this process, there will be a number of triplets that aren't required to create a protein, and they may just get in the way. So in RNA processing, we have introns and extrons. So the introns are going to be snipped out, And then the things that are remaining have to be put back together again, kind of like if you ever defragment your hard drive. So you remove the introns and then the exons are all spliced back together. In your text next to figure 312, you'll see that there's a Smart Art barcode that you can scan with your phone. All you need to do is take your phone with your camera feature, and when you open up your camera, when you take the image of the Smart Art barcode into the focus, it should automatically ask you whether you want to go to this link online. So that's just something else you can do to help understand this. So this image here in your text is going to explain mRNA transcription. So this is only showing a single part of a DNA molecule, just a really small portion, and this gene is undergoing transcription. So in the very first step, you'll see that there are two DNA strands, and they're going to separate, and they separate just ahead of that RNA polymerase enzyme that you can see as the big blue blob in the image. So in the second step, that RNA polymerase is going to move from one nucleotide down to the other, moving along the length of the template strand, pretty much like a zipper. So it's separating out the zipper, if you will, as it goes, and then it's actually closing up back behind it. And then in the third step, what you'll see is when the RNA polymerase reaches the stop signal at the end of the gene, then that's it. The RNA polymerase and the mRNA strands detach, and then you have the two DNA strands that can reassociate. Let's look at the process of translation a little more closely. So this is really the final step of protein synthesis. And all that we're really doing here is we are converting messenger RNA into an amino acid in a polypeptide chain that can become a functional protein. So we remember that gene activation and transcription is going to start in the nucleus, and the messenger RNA is going to exit the nucleus via a nuclear pore. So once it arrives at a ribosome, we're going to have the beginning of translation. So let's start with image 1 here in figure 3-13. So this is the binding of a small ribosomal subunit to messenger RNA. So you can see the messenger RNA strand is coming in. It has a start codon, which indicates it's time to initiate translation. So the messenger RNA comes in, and there's also an amino acid, in this case is methionine, and it's coming in via transfer RNA bearing an anticodon which matches the start codon. So you can see here that A matches with U, and then U matches with A, and C matches with G, right? So this is the very first step. The second step is when we have a large ribosomal subunit, which makes the ribosome complete, binds with the small ribosomal subunit, and this encloses the strand of messenger RNA. So we can see on the large ribosomal subunit, there are 3 different letters indicating the transfer RNA binding sites, and these are called E, P, and A. Think Environmental Protection Agency. And these stand for E, exit, P for polypeptide building, and A for arrival. So the messenger RNA strand is going to arrive in the site called A, and then at that point it will meet with the first amino acid via transfer RNA with its anticodon. It then will push along forward into the second site, the P, which is the polypeptide building or binding site. Then next what's going to happen is we'll have a second transfer RNA arrive at the arrival site, A site on the ribosome, and it's going to carry a second amino acid. So as this chain moves along, what's going to happen in the stage of P, the polypeptide building or binding site, we're going to bind with a peptide bond those 2 amino acids together. And then the fourth step is we're going to be extending the length of the polypeptide chain. So the messenger RNA just keeps chugging along through the arrival site, the polypeptide building site, and then the exit site. And so in this way, hundreds of amino acids are going to be sequenced, and we're going to string together a number of these polypeptides to create a completed polypeptide chain. And eventually what this means is we will have some functional proteins we can use, like hemoglobin or collagen. A protein has 4 different stages that it goes through in order to become a fully functional protein. So first we have the primary sequence, which is strictly the amino acid sequence, as you see indicated in A below, which is just a linear chain of amino acids strung together one after the other. The secondary the secondary structure is going to be either an alpha helix or a beta pleated sheet. So you can see examples of what those look like below. The tertiary step is when the protein folds into a globular structure, as you can see in the image below. And quaternary, or the fourth, is when you have an aggregate of two or more proteins coming together to create a functional protein, such as, for example, hemoglobin, or we have keratin or collagen demonstrated below in D. Let's shift gears and transfer into diffusion. Before we go any further talking about the movement of molecules like diffusion and osmosis, let's talk about some factors that are absolutely imperative to these things taking place. So first off, we have something called a concentration gradient. And basically all we're saying here is that there's a difference in the concentration of anything between one side of the plasma membrane and the other. So for example, sodium. So we have, let's say, more sodium in our extracellular fluid than we do in the intracellular fluid inside the cell in the plasma membrane. So in this case, we would say that we have a higher concentration gradient that's high in the extracellular fluid and low in the intracellular fluid. Now, a pressure gradient isn't about concentration and the number of molecules. It's based upon pressure differences across the membrane. So that's a completely different thing, but the idea of a gradient is the same in that it's going to be higher in one place and lower in the other across a membrane. And so that pressure difference can help to push a substance in a certain direction. Now, an electrical gradient has the same effect of being able to push something across a membrane, but with electrical gradients, they can either attract or repel molecules because of the difference in the electrical charge. And then lastly, we have the more complicated electrochemical gradient. And so this is a combination of the electrical gradient and the concentration gradient together. So this is going to control movements of ions and molecules in our cells and is particularly applicable to neurons. We're going to discuss several different mechanisms of mediated and non-mediated transport. But before we get into that, I'd like you to understand the difference between what the two are. So a mediated transport is going to require a very specific protein to be involved. But a non-mediated transport doesn't require a particular protein carrier or channel. It's just going to proceed through the lipid barrier without anything helping it along. So a passive process like diffusion occurs like you see in the image below. So what happens is we're looking at a concentration gradient for sugar in this example where we have a sugar cube placed in a jar of distilled water. So the water is literally just H2O with nothing in it, and the sugar cube is just sugar. So when we drop that sugar cube into the water, what's going to happen is the sugar concentration in the sugar cube is very high, right? Because it's all sugar. And in the distilled water, since there's only water, the sugar concentration is 0, right? So concentration gradients are going to indicate that things will typically go passively from high to low. So the sugar cube is going to dissolve its sugar molecules into the distilled water until it's finally eventually equally distributed throughout the entire solution. That's the basic concept of diffusion. Lipid-soluble molecules are going to be able to diffuse directly through the plasma membrane, even though the plasma membrane is a selectively permeable membrane. So examples of this include things like cholesterol and steroids. However, most non-lipid soluble molecules, and ions too, they don't diffuse through the plasma membrane on their own. So some specific non-lipid soluble molecules and ions are going to pass through membrane channels or other transport proteins. We're going to discuss these coming up. Here's some questions to ask yourself about diffusion. So first off, which substance is moving? Is it water, the solute, or both? And in the case of diffusion, it is the solute that is dissolving, right? The sugar cube is dissolving into the water. So it's the solute that moves. And is it moving with or against the concentration gradient? Well, it's moving with the concentration gradient, going from the high level of sugar in the cube to the no sugar in the water, right? So it's moving down the concentration gradient where there's lots of sugar to where there's no sugar. And is there a carrier required? Well, this is a trickier question because we have something called channel-mediated diffusion. And this allows things to pass, like ions, for example, through the plasma membrane. And these channels are always open, and they're called leak channels or passive channels. So yes, technically a carrier can be required for facilitated or hydrophilic reactions, or the answer also could be no. So for hydrophobic reactions, we will not require a carrier for simple diffusion. And then is it mediated? Well, if it's a hydrophilic, yes, it is mediated, but if it's a hydrophobic substance, then no, it is not mediated at all. And then second to last, is ATP required? Well, there are no cells here, there's no mitochondria. No, this is just simply sugar dissolving into water, right? So in this case, ATP is not required. And the transport method, is this active or passive? Well, these are actually both passive mechanisms. So whether you're talking about simple diffusion or whether you're talking about channel-mediated diffusion, this is still considered a passive process. There are various things that affect the rate of diffusion, and you're going to be discussing this in your labs this week. So the rate of diffusion is going to be affected by temperature. So if you think about how molecules move around much more quickly when they're heated up, that moving around more quickly will help to dissolve that sugar cube in water. So also the concentration gradient is going to be affected too. So if the concentration gradient of sugar is very high and the water is very low, the sugar has lots of space to dissolve and diffuse very easily. Now by contrast, let's say the concentration gradient was lower. And what I mean by that is, let's say we drop a sugar cube into a cup of sugar water already. Well, in that case, there's plenty of sugar already dispersed throughout that sugar water. So it's going to be a little bit slower for that sugar cube to dissolve into the water. Also, there's an electrical gradient impact, so that can either facilitate or slow down the diffusion rate. So if the electrical current is active, it can promote the rate to increase. Also, molecular size is a factor that affects diffusion. So as you can imagine, smaller things will diffuse more quickly, larger molecules will take a little bit more time. And then lastly, the distance to be traveled. So if the sugar has to travel very far in order to dissolve, that'll take more time, right? But if it can just dissolve into the immediate H2O molecules around it, that can happen much more quickly. Here's a great video that shows you how osmosis works. Now keep in mind, osmosis refers only to water and how water changes its concentration, not the sugar. When we talk about the sugar dissolving into the water, we're talking about diffusion. When we talk about water moving, we're talking about osmosis. Osmosis is a passive process that applies only to the movement of water. So in figure 316 from your text, which you can see below, you'll see an example of water and a solute together in a U-shaped tube. And down at the bottom, there's a selectively permeable membrane that will only allow water to pass between it, okay? So if you look at step 1, what you see is all those little blue dots are water molecules, and there are plenty of them over on the A side, but then on B side, you can see there's considerably fewer water molecules. So if water is going to osmotically move from an area of high concentration down to low concentration, down the concentration gradient, it's actually going to move a lot of water from column A over into column B. The result of this is that the volume of column B is going to go up because water is going to create a lot of space, right? It takes up a lot. So the volume increases due to this selectively permeable membrane. And the volume will decrease in A as more water osmotically moves over to column B until the pressures are equal on either side. Now, there's one way to stop this and keep one side, for example, column A that has tons of water and column B which doesn't have much. There's a way to keep these the way they are, and that is by applying force on the area with the lower concentration gradient for water. So you can see in step 2B, they put a little stopper on the top of column B, and because that column can't allow the fluid to rise anymore, the water is not going to be able to go over anywhere because there's literally no space. So here's some questions to ask yourself about osmosis. So first off, which substance is moving when we discuss osmosis? Is it water, a solute, or both? Well, as you just saw in the previous slide, it is only the water that moves. It was not the other molecules. So it is only water that moves, and it moves across a semi-permeable membrane. For example, our plasma membranes. The next question, which happens with a concentration gradient? Does the substance move with or against the concentration gradient? As you saw from those two columns of water, it moves with the concentration gradient of water. Technically, this also is against the concentration gradient of the solute. Next question, is there a carrier required, yes or no? Well, no, there's no carrier at all. This just happened independently on its own. Was it mediated? Again, no. And was ATP required? Well, again, this is just sugar and water. These are inert substances. There's no cells doing anything. There's no mitochondria generating ATP. So the answer is no. And then last question, what is the transport method? Is this active or passive? And this is a passive process. It happens on its own independently. Another passive process, just for your notes, is called filtration. And filtration is non-mediated, and it is the movement of both a solute and water. And so this is based strictly on a pressure gradient, not a concentration gradient. So the best example of this is going to be the kidney. So the kidney relies on filtration to produce urine. When we discuss molarity, we're really just discussing what measurement there is of an amount of a certain substance. So we use Avogadro's number, which is 6 times 10 to the 23rd power, to be able to calculate this. When we talk about osmolarity or osmotic concentration, what we're specifically referring to is what is the total solute concentration in an aqueous or water-based solution. And now the number of particles that are present in that solution can affect the rate of transport. So larger volumes of particles are going to be slower and smaller volumes are going to be quicker. When we refer to the effect of a fluid on a cell, we're not going to be talking about osmolarity in some sort of general chemical sense. We're going to talk about tonicity because this is what really matters. It's how does this fluid affect the cell and our tissues in our body. So we'll have 3 different ways to describe this. It can either be isotonic, hypotonic, and hypertonic. So in the first example, what you see is a red blood cell. You can see the biconcave disc there. It looks totally normal. And the extracellular fluid, in this case, has the same concentration as the intracellular fluid. So because those are the same, the cell doesn't change. There's no change whatsoever to its shape or anything. We call this isotonic. Now, if you move one over to the middle image, you'll see that there's a cell that's swollen up. And the reason it's swollen up is because this is a hypotonic solution. So there are fewer solutes in the outer extracellular fluid than there are in the intracellular fluid. So the water molecules will rush into the cell and eventually cause it to burst and lyse. Then lastly, on the right, we have a hypertonic solution in which there are tons of solute molecules outside of the cell and then fewer inside. So what happens is water will actually exit the cell to dilute the extracellular fluid. And as a result, this will shrivel or crenate the cell. So just to reiterate, in an isotonic solution, the isotonic solution has the same osmolality as the cell. So there is no net movement of water or a solute, and the cell totally remains normal looking. In this case, we have a red blood cell looking like a normal biconcave disc. In a hypotonic solution, you're going to have less solute or lower osmolality in the extracellular fluid than in the cell. So the net water movement is from the extracellular fluid into the cell, and if the solute can leave, then it will, but that's not necessarily always the case. And what we end up with is massive swelling. Again, this is the water balloon example. So you end up with something called lysis possibly, and in red blood cells we have a particular name we apply to this, and it's called hemolysis. In a hypertonic solution, we have an extracellular fluid with more solute or a higher osmolality than in the cell. So the net water movement is out of the cell, so water will move out to dilute that extracellular fluid, and if possible, the solute will move into the cell. But in so doing, the cell is going to shrivel up, shrink, and crenate through a process called crenation. Here are a couple sample questions for you to see if you're on track. So a hypotonic solution will have blank than the solution or cell used for comparison. Answers are A, less water and more solute, B, equal water and solute, or C, more water and less solute. So you can tell right away that options A, B, and C are, in no particular order, isotonic, hypotonic, and hypertonic answers, right? So we're just asking, what really is a hypotonic solution? And the answer is C, it's going to have more water and less solute than the solution or cell used for comparison. So here's one where you have to think a little bit. So here's the pretext. Solution A is hypertonic compared to solution B, and therefore solution B is hypotonic, in reverse, compared to solution A. So the question is, if solutions A and B are separated by a selectively permeable membrane, water will move from, and you have options A to B or B to A. So think for a moment about that example of osmosis that I showed you with the two columns in that U-shaped tube, right? So we had that selectively permeable membrane in the center that only allowed water to pass. So if we have a selectively permeable membrane, water will move from where to where? Well, if we have one solution that is hypertonic and one that is hypotonic, it's going to move down the concentration gradient from the hypertonic solution to the hypotonic solution. So the answer is A. The solution will move from A to B. So here's a little bit more tricky question. In a hypertonic solution, what will happen to the cell if a plasma membrane is not permeable to the solute? So we have a concentration gradient that is higher in the solution than in the cell, but the membrane won't allow the solute to pass. So just because the solute can't pass through the membrane doesn't mean that water can't pass, right? So water is going to go down its concentration gradient via osmosis to make things more level. So if we have a hypertonic solution, so let's say a high sugar solution, we drop a cell in it, the sugar can't go anywhere, but the thing is water can. So water will leave the cell to go into that fluid to dilute it. So in that case, the answer is D, the cell will crenate. Okay, here's one last question. In the following example, what is the term used to describe the solution surrounding the cell? So is it hypertonic, isotonic, or hypotonic? And in the example, we have a schematic showing you a cell that is 0.9% salt, and the solution is 1.7% salt. So right off the bat, we can rule out B, right? Because we know it's not isotonic, because in that case, we would have the same percentage in both the fluid and in the cell. So we know it's either hypertonic or hypotonic. So in this case, we have a higher concentration gradient of salt in the fluid than in the cell. So because of that, we are going to call the solution hypertonic, A. Let's talk about membrane transport. It's section 3-6 in your text. So what is microtransport? Basically, this is just the transport of small molecules across the plasma membrane, which you'll remember is a semi-permeable membrane. So the plasma membrane is going to regulate this microtransport, allowing some things in and others not. And this will occur up until the point where equilibrium is reached, which is our steady state of homeostasis that we're looking for. But keep in mind that equilibrium doesn't necessarily mean that we have exact equal concentrations in both places. So the microtransport processes include diffusion, facilitated diffusion, osmosis, and active transport mechanisms. So what is the difference between a passive and an active process when we're talking about microtransport? Well, a passive process doesn't require any ATP at all, right? It just happens on its own passively and it moves with the concentration gradient. So 3 examples of this are diffusion, osmosis, and facilitated diffusion. Active processes, on the other hand, are going to require ATP or another energy source in order to make this happen, because we're moving substances against the concentration gradient. So examples of this include antiport, a symport, or bulk transport. Mediated transport is going to require some sort of structure in order to facilitate this process. So a channel, a carrier, or a pump is going to help move a substance across a semi-permeable membrane. So characteristics of these channels, carriers, or pumps are the following. So it's going to be specific, meaning that a specific channel, carrier, or pump is only going to have a certain molecule that it will allow to pass. Also, saturation is important. So if there are 5 sodium channels, let's say hypothetically, then only 5 molecules can come through at a time, 1 in each of those channels. So it doesn't matter if there's 5 million sodium molecules on one side, they can only come at a certain rate based upon the saturation of the carriers or channels. Regulation is another, and so this is important because chemical messengers are going to be present to help open up these channels or to activate the carriers. So there's this external regulation occurring. So for example, the glucose transporter is going to be stimulated by insulin, which we described earlier. And then last, competition. So there's going to be competition between similar molecules that might be able to go through the same carrier, space, or pump. In facilitated diffusion, what we have is a carrier protein in the selectively permeable plasma membrane, which will allow something like a glucose molecule to be able to come into that specific carrier, and then the carrier is going to adjust its shape to close so that way nothing else can come in while that one glucose is being transported into the cell. So now please keep in mind that although this looks like a unidirectional process, that this can actually reverse and have the glucose molecule be ejected from the cell if the concentration gradient reverses. So here are some questions to ask yourself about facilitated diffusion. What substance is moving? Is it a water, or is it a solute, or is it both? And in this case, we're talking about solutes. And are we going with or against the concentration gradient? Well, in this case, we are going with the concentration gradient regardless of which direction that is. Whether it's a high concentration gradient on the extracellular fluid or an intercellular fluid, we are going from high to low. Is it carrier mediated? Well, yes, we have the carrier protein that we just looked at in the image on the left. Also, is it mediated? Yes or no? Well, yes, it's mediated. And is ATP required? Well, no, there's no ATP required in this process at all. And then lastly, as a transport method, is this active or passive? This is actually a passive process. Even though the carrier protein is facilitating it, it happens passively. So keep in mind that glucose is an incredibly important substance in our bodies. So if you'd like to learn a little bit more, here's a video for you to review on your free time. A great example of primary active transport is the sodium-potassium pump, of which you will never hear enough of. And this is an ion pump. And so for every 3 sodiums that it pumps out, it's going to bring 2 potassiums in. So this sodium-potassium adenosine triphosphatase is an enzymatic action that's involved in the process. And this is a required enzyme because both of these molecules are moving against their gradient. So the pump is physically forcing them against the concentration gradient. Secondary active transport is kind of interesting. So this is powered by the diffusion of another ion or molecule, and usually this is sodium. But the molecule is going to be moving with the concentration gradient, and this is going to provide the power to carry another molecule along for the ride. So somebody is more or less hitchhiking. So in the example that you see below, you'll see that glucose is coming along with sodium. And so as this happens, then later on, it's possible in secondary active transport that while no adenosine triphosphate or energy is needed in this process, there may actually be adenosine triphosphate needed in the future to pump something out of the cell. Another interesting point is symport and antiport. So if you have a second molecule that's moving in the same direction, we call that symport. But if you have a second molecule at the same time through the same channel moving in the opposite direction, we call that antiport. Which type of transport moves sodium against its concentration gradient at the same time as potassium is being moved against its concentration gradient? Your options are A, primary active transport, B, cotransport, also known as symport, C, antiport, or D, facilitated diffusion. Well, we can rule out facilitated diffusion right away, and cotransport would have 2 things moving in the same direction. Antiport is a tempting option because we have 2 substances moving in different directions. But because we're having these substances both move against their concentration gradient, we are thinking immediately of the sodium-potassium pump, which gives you the correct answer of A, primary active transport. Bulk vesicular transport is very different than the transport of molecules just on their own. So in this case, we're moving big substances across the plasma membrane. So because we're moving something big, it's an active process which requires adenosine triphosphate. There is no carrier involved in this, and it may require receptors to help capture the molecule. So there are 2 different processes here that we're going to discuss, endocytosis and exocytosis. So endocytosis is moving something into the cell, and exocytosis is moving something big out of the cell. There are 3 different types of endocytosis, of transporting bulk vesicular substances into the cell. So there's phagocytosis, pinocytosis, and receptor-mediated endocytosis. So phagocytosis literally translates to cell eating. So the cell is actually consuming a considerable chunk of some sort of substance. Pinocytosis is cell drinking. So in this case, of course, we're talking about a fluid more than a solute or substance. And receptor-mediated endocytosis is going to require a receptor to bind to the correct molecule to make this occur. In exocytosis, we are moving by bulk products out of the cell. So these vesicles are going to come from the Golgi apparatus and migrate towards the plasma membrane. Following this, it fuses with the plasma membrane and then bursts to release its contents out into the extracellular fluid. Following this, the plasma membrane of course repairs itself and maintains its integrity. Phagocytosis occurs when a cell actually consumes a substance in the extracellular fluid. So this is something that you can imagine happening oftentimes when we talk about macrophages and other parts of the immune system. So something called pseudopodia, which literally translates to false feet form. Now, in my opinion, it should have been something like a false mouth, because really what's happening is the cell is opening up, creating a mouth, and then surrounding that particle and enclosing it inside the cell. So then the membrane will fuse along behind it, and it forms a vesicle within the cell. Pinocytosis is similar to phagocytosis, but because we're talking about fluid and not consuming a substance, we don't need the cell to actually develop those pseudopodia and surround anything. All that has to happen in pinocytosis is the plasma membrane needs to create these little invaginations, so that way the fluid can literally just spill in. And then once the fluid has spilled in, the membrane can fuse back again, and the vesicle forms, and it's brought that fluid in bulk into the cell. Receptor-mediated endocytosis is a process that will bring certain contents into the cell in a very high concentration. And they do this by having ligands that will bind to specific receptors that are in these clathrin-coated pits that you see as number 1 in the image. And so what will happen is after these ligands have bound to the receptors in the clathrin-coated pit, is that this pit is going to basically balloon out and then close in to create a completely sealed coated vesicle within the cell. So that coated vesicle will then fuse with a primary lysosome, become a secondary lysosome, and that secondary lysosome is going to be responsible for removing those ligands and then basically sending any byproduct back out through a process of exocytosis to be removed from the cell. Now let's talk about section 3-7, membrane potential. Section 3-7 in your text discusses membrane potentials, and really all this is discussing the difference between the positive and negative charges across the plasma membrane. So that's basically exactly the definition of a potential difference. It's the plasma membrane separating a positive and negative charge. But then when we talk about that exact same potential difference in reference to a plasma membrane, we now call it a membrane potential. So this is the potential difference across the plasma membrane. And then if we look at a plasma membrane of a cell at rest, we would call that a resting membrane potential. We're going to discuss this in a whole lot more detail as we move into the nervous system. Here's a pop quiz question for you. Certain cells in the ovaries and testes contain large amounts of smooth endoplasmic reticulum. Why? To be able to answer this question correctly, you'll need to know what the smooth endoplasmic reticulum does. And in particular, the tip-off is that ovaries and testes are involved. The answer is D, to produce large amounts of steroid hormones. Please take a moment to review the functions of the various organelles to be able to know that you can rule out options A, B, and C. Let's talk about section 3-8, the cell life cycle. Mitosis is the process by which cells divide to produce 2 identical daughter cells that each have 46 chromosomes, or 23 pairs, which is a full complement. Meiosis is a process of special cell division in which we're not creating somatic cells throughout our body, but rather we're creating gametes, which are sex cells for reproduction. Cells spend most of their lives in interphase, which is a resting period of normal cell life. So interphase has to occur before mitosis can begin, which will include prophase, metaphase, anaphase, and telophase. So before those things can begin, interphase has to go through the following process. So it'll spend most of its time in G0, which is normal cell life. In G1 of interphase, we will have replication of centrioles and organelles, and this is in preparation for division. Then it'll move into stage S, which is replication of DNA, and G2 includes protein synthesis. At this point now, the cell is ready to begin mitosis. In a resting cell, you'd expect to see the genetic material in the nucleus as a tangled mess of chromatin, but in prophase, the chromatin is going to condense and start to form chromatids, which are that normal X shape that we would expect to see in a karyotype. So the centrioles are very important because these are going to migrate to opposite poles of the cell, and they spin spindle fibers, and these all help to organize all those chromatids in the nucleus. The nucleoli and nuclear membrane are going to dissipate in preparation for spreading of this genetic information. In metaphase, the chromatids line up on the equatorial plane. So you can see all those X's lining up right down the center of the cell and the center of the screen, and spindle fibers attach to centromeres, which are now oriented at both ends of the cell. In anaphase, the centromeres will split, and the chromosomes are pulled and migrate towards the poles. You'll see on one side of the cell that there's a cleavage furrow, which is just an indentation, and this shows you where the cell is about to separate once cytokinesis occurs. So cytokinesis is beginning, but it hasn't fully occurred yet. In telophase, the chromosomes will unravel, and the nuclear membrane and the nucleoli reassemble so that we are about to have two totally separate cells. So cytokinesis continues, and at the end of telophase, we have cytokinesis complete. Cytokinesis literally translates to cell movement, so this is indicating that the cells have completely separated. Finally, cytokinesis occurs, which marks the end of cell division. Two totally separate and complete cells are ready to go, and they each have their own plasma membrane. The mitotic rate is really just the rate of cell division. So a faster mitotic rate means a shorter cell life, whereas a longer mitotic rate means a longer cell life. Cell division requires energy in the form of ATP, and every time a cell divides, there's a potential for something to go wrong. And if something goes wrong in that cell division and there's a mutation, it could result in a malignancy. Muscle cells and neurons rarely divide, which is one of the reasons why these tissues are more difficult to repair. There are some cells in our body, like skin and digestive tract cells, that only live for a few days or hours, and they have to do this because they're constantly assaulted by things from our environment. So these cells are constantly replenished by stem cells, which are unspecialized cells that are capable of differentiating into various cell types. Stem cells are present in the umbilical cord blood of newborns and are also present in embryos, which is what allows two individual cells of haploid nature to be able to replicate and to become an embryo and to continue to develop and differentiate into all the different cell types of our body, ranging from our skin to our liver to our spleen. So, pop quiz question, what would happen if spindle fibers failed to form in a cell during mitosis? Well, we know the spindle fibers are an important part of pulling our genetic material to the side, so that way we could have that cleavage furrow and separate into two daughter cells. So if we had the spindle fibers failing to form, we would end up with chromosomes that would not separate. C. Let's discuss cell cycle regulation from section 3-9. Cell life cycle regulation is fascinating, but only covered very briefly in this chapter. So here's what you need to know. MPF, or M-phase promoting factor, also known as maturating phase promoting factor, is the main trigger for cell division. There are a number of different growth factors, and these are just chemical factors that affect cell division. So here are a few examples. Prolactin, for example, is produced by the anterior lobe of our pituitary gland, and it's important for stimulating cell growth, division, and development of mammary tissue. So a newly postpartum mother will be able to have her mammary tissue mature fully to produce milk because of prolactin. Also, erythropoietin is another important growth factor, and it's produced by our kidneys. Its effect is to act on the red bone marrow to stimulate the development of more mature red blood cells. And then also, growth hormone, you've probably heard of that before. It's pretty important, and it's produced by the anterior lobe of our pituitary gland also. That's a very common site for a lot of these to be produced. And it's going to affect pretty much all cells, but particularly epithelial and connective tissues. Telomeres are terminal segments of DNA that are going to regulate the number of cell divisions that a cell can complete. And then the last thing that I've listed here is the Hayflick limit, and this is not testable material, but I thought you'd be interested. The Hayflick limit is a phenomenon that's been recently discovered to basically decide what the number of times is that a cell can divide before it enters senescence. Because we don't live forever, and after a while our cells are not going to continue reproducing. We have a finite lifespan, and the Hayflick limit is that exact calculation that figures out how many times can a cell divide before it will not divide anymore. Okay, so now we're getting into my neck of the woods with abnormal cell growth. This is section 3-10 in your text. A neoplasm is the technical term for any tumor or new growth, whether it is benign or malignant. Benign neoplasms are not cancerous. Now, they can still become very large, but what they do is they compress upon adjacent structures rather than invading them. Now, malignant structures, these are what we would call cancerous, and malignant structures will invade to local tissues and also distal tissues. And so the invasion is the key point that differentiates benign from malignant tumors. And also, malignant tumors are not responsive to the normal cellular controls, and these cells have a very high energy need. So you can define cancer as an abnormal proliferation of cells anywhere in the body. So just for fun, I wanted to show you some images of some gross specimens and have you guess whether you think these are benign or malignant. So we have specimens here, 1, 2, 3, and 4 going clockwise, starting from the top left. And just in your head, take a look at each of these and think about whether or not you think they're benign or malignant. Now, these are all sort of rounded, smooth, contained lesions, and even the one in the bottom right-hand corner, which looks kind of bizarre, it's contained by a membrane. So all 4 of these are all benign lesions. Here are 4 more images for you to consider. At the top left, we have a uterus that's been bivalved, revealing the endometrium. And as you can see, there's this exophytic mass just spilling out everywhere, invading into the endometrial tissue. At the top right, you're going to see the exterior of an ovary that's very ballooned out. It's very large. But of note is you'll see that there's this pink, grainy, papillary structure that's at the bottom right of it. And then if you go down to the bottom right, you're going to see a cross-section of bone, and what you're going to notice in there is a whitish area, but there are also areas that are kind of brown and pink and irregular, and it's kind of hard to differentiate where exactly the tumor starts and stops. And then at the bottom left, you're going to see, this is zooming in on a section of open bowel. So you see the folds on either side of the image that are normal, and in the center you see a big round lesion that's taking up the majority of the image. So all 4 of these are malignant. Now, it's not as simple as saying, hey, if it looks nasty, that's malignant, and if it looks more rounded and intact, it's benign. In fact, there are many things that go into making the differentiation between a benign or malignant tumor. And in fact, some of these differentiations are only made microscopically. But nonetheless, you kind of get the idea that the malignant ones are the cancerous ones, and they're obviously nasty, and they're obviously going to be very invasive. So neoplasia is defined literally as a new growth. So characteristics of malignant neoplasms are the following. So they have uncontrolled autonomous cell replication. And so one of the ways this is commonly described, it's as though the cell nucleus has been hijacked. So it's replicating in a totally unusual form. They also lack normal cell apoptosis, which is programmed cell death. So at a certain point, cells die and the new cells come up to replace them. But in a cancerous lesion, these cells don't just die, they just keep growing and growing and growing. So they basically live forever like vampires. Also, they have an unlimited potential for cell replication. They'll just keep going and spread throughout the entire body. They're insensitive to anti-growth signals, and they also evade the immune system, and that is key. And this is also a key in some of the newer modalities of treatment for cancer. Further, neoplasias, specifically, not just benign neoplasias, but malignant neoplasms, are going to have an entirely new vascular supply that they request themselves. They actually release chemicals into the surrounding tissues that will stimulate blood vessels to grow towards them, so that way they can obtain their energy needs directly from the bloodstream. And this is a process known as angiogenesis. They locally invade adjacent tissues, and what I mean by that is they're not just pressing up against the adjacent structures, but they're actually having finger-like extensions that reach into the tissues and can interrupt their normal function. And the other thing they do is they metastasize. So metastasis is the state of migration of a neoplastic cell to an abnormal location. So you've probably heard this before. So if somebody has cancer of the colon, a very common site for it to metastasize to is the liver. Now, this is not 2 primary cancers, so they're not 2 independent cancers. You have one cancer, which is the colon cancer, but through metastasis, it is spread to a different part of the body. And so it has the same biological markers, but it just happens to now also be in the liver. So that cancer that started in the colon can spread to the liver and also the brain and a number of other places as well before it finally eventually will kill the person without treatment. A mutagen is an agent that causes a mutation in the DNA, and a carcinogen is any type of agent which may be a mutagen, but it will cause cancer. So malignant transformation is one of those things that takes multiple hits. So you can sometimes have a genetic predisposition to potentially develop cancer, but then when you accumulate a mutation through the environment and your genetic background, that can lead to the development of a malignancy. So your risk of cancer in general is going to increase with age, with the effect of various hormones, and also with the inability of your body to clear free radicals from it. Oncogenes are genes which are going to increase growth when mutated or upregulated by various substances. And tumor suppressor genes, when they mutate, will also allow for increased cell growth. This schematic here indicates the mechanism of hematogenous metastasis, which is cancer spread to a different location through the bloodstream. The other mechanism is lymphatic, which is moving through the lymphatic system, which is why when people have cancer, we typically do a lymph node dissection and resection as well. So what you can see here is it starts off on the top left with an abnormal cell, which is indicated in purple. And so if you move to the second image over in the right, after a series of cell divisions, that one abnormal cell, which has basically been hijacked and gone totally crazy, is now replicating itself irregularly with no regard to normal rules of where it should grow and how long it should live. So it's going to extend into all different places that it shouldn't locally, and when it gains access to the bloodstream, a couple of these unusual cells will circulate through the bloodstream and through different breaks in the vascular supply will ooze through the walls of the vascularity and move into a secondary location, creating a mass of secondary tumor cells known as metastasis. This slide is totally untestable, but it bridges our discussion of neoplasia with the tissue level of organization, which is what we're going to discuss in the next lecture. Actually, I always include this in my labs because I think it's really important information and helps to drive home the points of the way that our cells are oriented. So if you'll bear with me, this is the Bethesda system, which is a way of grading cervical pap smears to see if they are cancerous or not. And as you can see, this is a continuum. So we start on the left with normal tissue, and over the far right, we have invasive carcinoma. And what you can notice is a diagonal line that basically runs from left to right heading up. And this is showing you that the tissue on the far right, with all those big cells that have the big nuclei and very little cytoplasm, those are all abnormal cells that have been hijacked by HPV strains. Whereas the cells up at the top that are poofy with just tiny little nuclei, those are normal cells that have matured appropriately the way they should. So what differentiates carcinoma in situ from invasive carcinoma is the ability for those cancerous cells to break through the basement membrane. So this is the same sort of thing that you'll see if you're talking about ductal carcinoma in situ versus full-on breast cancer. The exact same thing is true. In ductal carcinoma in situ, you have full thickness involvement of these abnormal cells, but it hasn't broken through the basement membrane yet. Breaking through the basement membrane takes a lot of energy for a malignancy. So when it breaks through the basement membrane, it officially gets qualified as an invasive carcinoma. Here's another pop quiz question. During DNA replication, a nucleotide is deleted from a sequence that normally codes for a polypeptide. What effect will this deletion have on the amino acid sequence of the polypeptide? And your options are A, no effect, the deletion will be skipped. B, no effect, the deletion will automatically be repaired. C, the amino acid sequence will disintegrate. Or D, the amino acid sequence would be altered. Well, the first three are all things that we could not possibly know, but we certainly know that the amino acid sequence would be altered. So the correct answer is D. All right, lastly, let's talk about cellular differentiation, which is 3-11 in your text. Stem cells contain all of the genetic information to create all of the different types of cells in our body. And all of the somatic cells in our body are going to contain the same chromosomes and the same genes. Cells undergo cellular differentiation by turning off genes that are not needed by that particular cell. So for that reason, our columnar epithelial cells, our smooth muscle cells, our bone cells, etc., they all contain the same chromosomes and the same genes, but they each turn off different genes that aren't needed in order to become different cells. Lastly, I want to thank you for staying with me and listening to the very end. Thank you so much. This is important information. I know it feels abstract, but this is the very foundation of what we're going to be discussing in the subsequent chapters. You definitely need to know this and understand it in order to be able to be successful both in the rest of the class and in your subsequent classes. So again, thank you. I'm here for you. Please contact me if you have any questions whatsoever, and best of luck. Stay healthy.